Thank you all for coming to our second Ross Memorial Lecture Series of this semester. It's my great pleasure to introduce Dr. Ashley Helton, who is an assistant professor in the Department of Natural Resources and the Environment at UConn. I first met Dr. Helton a couple of years ago, I think probably when you were really new to Connecticut. Brand new. When we were both asked to sit on a study group put together by the Connecticut Academy of Sciences and Engineering. They were doing a study on ways of managing phosphorus pollution. And so we were both asked to sit on that committee. It was kind of my first foray into management side of things. It was a very interesting experience, but one of the benefits of being on that committee was as I met Dr. Helton and started to learn a little bit about her work. So we're thrilled that she's here with us today to talk to us about some of the work that she's doing. Dr. Helton has an undergraduate degree in environmental studies from the University of Cincinnati. She then went on to University of Georgia, where she got her master's and Ph.D. in ecology, and then did a postdoc at Duke, and now, as I mentioned, she's an assistant professor at UConn. So please join me in welcoming Dr. Helton.
Thank you. Well, thanks. Thanks for inviting me to give a seminar. I'm really excited to be here, and I've gotten to hear about a lot of cool things going on at Fairfield University. If you can't tell from the accent, I'm not from around here. I'm from Kentucky originally, grew up in the South. So this is my third Connecticut winter. This one is way, way better than the last two. The first winter I was almost convinced that maybe I shouldn't live in Connecticut. I bought my first snow shovel, which was interesting. So today I'm going to talk about how climate change, specifically sea level rise and saltwater intrusion, affects the capacity of freshwater wetlands to cycle carbon and nitrogen. But first, I guess I don't have to, I can use the pointer to click because we're remote. Oh, it should. Okay, so before I start talking about wetlands, I want to give, I guess, a little bit of background about myself and the type of research my lab does. So when I was trying to figure out how to do this in this presentation, I did a really fun thing where I Googled the first research project I was ever part of, and I found these great old pictures of myself, which is fun. So the first research project I was part of was an undergraduate research project in the Department of Geology at the University of Cincinnati. The title of it was Replacing Limestone with Dolostone in Anoxic Limestone Drains Eliminates Clogging with Gypsum. So a lot of geology words there, but it was really a project about how we can we effectively treat acid mine drainage to improve the performance of constructed wetlands that we used to treat this acid drainage that was just killing these constructed wetlands. So this is me circa 2001 or 2. I was a junior. And my first series of lab experiments, a column experiment and a batch experiment, a series of batch reactors to kind of test this idea about treating acid mine drainage. So I got this position by applying to a research program for women in science and engineering as an undergrad. I thought at the time I really wanted to be in the biology or the ecology department studying community ecology. And so I listed my first 3 choices as, plant ecology, stream community ecology, and all these labs I thought I wanted to work in. And then I got assigned to a geology lab. And so I was so disappointed that I got assigned this geochemistry project, but I thought I wanted to be a community ecologist. So I did it anyway, and it turns out that I really love chemistry. And chemistry has really influenced everything I've done since then. So I went on after the University of Cincinnati, like Jen said, and got my MS and Ph.D. in ecology, but both of these degrees were really in biogeochemistry. I really took that geochemistry project and incorporated it into all of the ecology that I've done throughout my academic career. I'm one of those weird people that went straight through school and took no breaks because I like to be in school, and now I'm still in school and I'll be in school the rest of my life. So I did a quick postdoc at Duke University, and now I'm at University of Connecticut as an assistant professor in the Department of Natural Resources, but with a joint appointment in the Center for Environmental Sciences and Engineering. So I have the longest title ever. So Anyway, that's kind of how I got to biogeochemistry. But, oh, right. And so my lab now does kind of a really wide range of biogeochemical research. So we're interested in how carbon and nutrient cycles interact and how human activities influence the way those cycles interact, things like land use change and climate change. We work using a variety of approaches. We do lab work, we do field work, we do simulation modeling, and we do this across a variety of freshwater ecosystems. So we work in streams, rivers, floodplains, and wetlands. Probably lakes are the only systems that we've steered a little clear of. I don't have much background in lakes. But this is my website, and you guys can read more about what we do, and particularly the really cool things that my students are doing, since they do most of the cool research now, at my website, which is ashleyhelton.weebly.com. Okay, so today, though, I'm going to talk to you guys about a couple of specific projects. I'm going to talk about wetland biogeochemistry. I'm going to spend quite a bit of time giving you some context and background, and so I apologize if this is a little bit too much background for some, but just stick with me. We'll talk about wetland ecosystem function, particularly the functions that my lab is interested in studying. I'll talk about what I mean when I say wetlands at the leading edge, the environmental stressors that are affecting wetlands, particularly coastal wetlands in the East Coast. And then I'm going to give you guys an overview and some results from two current projects. One in the southeast, on the southeastern coastal plain, and one here in Connecticut, on Connecticut's coast, that's just ramping up. Actually, one project that's kind of ramping down and one that's ramping up. So old and new. Okay, so coastal wetland ecosystems provide a really wide range of ecosystem services to humans. So they dissipate energy from storms and they reduce flooding, and we've all heard a lot about that in the media lately with increasing hurricane intensity and frequency. Coastal wetlands also prevent erosion. The plant roots hold on to soils. Coastal wetlands provide a lot of habitat for wildlife, birds, fish. Coastal wetlands provide a lot of recreational opportunities for us. We hike, we boat, we fish, we hunt in wetlands. And then coastal wetlands also play a huge role in improving water quality. So wetlands reduce excess nutrients and contaminants that enter them. They reduce their flow to downstream systems. Coastal ecosystems can be particularly sensitive to that kind of pollution. We all live in the Long Island watershed, so hopefully we all know that elevated nitrogen loads cause the seasonal hypoxic or dead zone events that we see in Long Island Sound. And then finally, coastal wetlands also store a large amount of carbon in their plants and soils. So they store so much carbon, in fact, that if you scale that up to a global scale, it can actually buffer climate change. So they're really important ecosystem services. So in our lab, we're particularly interested in those services that are mediated by biogeochemical cycling. In this case, carbon and nitrogen cycling. So because wetlands lie at this kind of transitional boundary, so wetlands lie kind of in the middle of uplands and of flowing waters, they receive really large loads of contaminants. So those contaminants can be nitrogen and phosphorus from fertilizers or other contaminants from urban runoff like heavy metals. So wetlands are particularly efficient at removing these contaminants through plant uptake, through microbial uptake or transformations, and even through chemical processes, physical processes like sorption to soils. So water with high contaminant loads enters wetlands, and then wetlands discharge water with much lower contaminant loads. So the first point here on this slide, I'm going to make kind of several, instead of walking you through the entire complicated world of wetland biogeochemistry, I'm going to make 3 points that are relevant to what I'm talking about today. So the first then is that wetlands are efficient at removing and retaining nitrogen in particular is what we're interested in today, but also other contaminants like phosphorus and metals. Okay, so second, wetlands are highly productive ecosystems. So that means that they produce plant biomass at a rapid rate. So in wetlands, the decomposition of that plant biomass after the plants die is really limited, and I'll talk about why in a minute. But because of that, all of that, or a large amount of that dead plant material, builds up as soil organic matter. So plants take up carbon dioxide from the environment, and because of their rapid growth and the limited decomposition, a lot of that carbon is buried and stored long-term within wetlands. And like I said in the previous slide, that's really important on a global scale because it can actually buffer some of those greenhouse gases that we release in the atmosphere. It can buffer global climate change. So the second point is wetlands are highly productive, and that's important because they sequester a large amount of carbon in their plants and soils. So a minute ago I said that decomposition was limited in wetlands. And so the reason that's the case is because of oxygen limitations. So oxygen diffuses about 10,000 times more slowly in water than in air. So when an area is inundated, the oxygen doesn't get that far, and steep redox gradients develop near the soil surface. So what that means is the microbial community that usually uses a lot of oxygen to decompose that plant materials doesn't have oxygen available. So it turns out that when microbes look for what we call an alternate electron acceptor to respire with, right, something in place of the oxygen to break down organic matter, the next most efficient thing is nitrate, which is a really important form of reactive nitrogen. So nitrogen is reduced by these processes mostly to dinitrogen gas. And so dinitrogen gas is fantastic because it's inert, right? It's not a greenhouse gas. It makes up about 80% of what's in this room. We're all breathing in it right now. So this is really the only permanent way that nitrogen can be removed from a biologically active pool to the atmosphere where it's not biologically active. Now once it's in the atmosphere, that nitrogen, N2, has what's called a triple bond, which means it's really, really, really strong, and it takes a lot of energy to break that. So once it's in the atmosphere for a long time. Now, the flip side of this is these anaerobic processes that happen in the absence of oxygen, also create greenhouse gases. Greenhouse gases like nitrous oxide and methane. Nitrous oxide is about 200 times stronger than carbon dioxide as a greenhouse gas. Methane is about 25 times stronger than carbon dioxide as a greenhouse gas. So the third point is that wetlands create an environment for these anaerobic microbial processes that are really important in their ability to permanently remove nitrogen, or as permanently as you can, right, on a medium time scale, but they also produce greenhouse gases. So when we're understanding the services that wetlands provide and how they function, it's important to understand the balance between removal of pollutants and production of greenhouse gases. Okay, so those are kind of the three points we're going to focus on today. And in fact, I lost some of my animation. In fact, I had colors. In fact, we're going to focus really on two of these points most of today, and that's 1 and 3 here. Wetlands as efficient nitrogen removers, and the balance of anaerobic processes in greenhouse gas production. Okay, so thought I might need my notes for that one. It was going to be kind of complicated. I didn't use them all that much though. So hopefully that means it wasn't gibberish. So I'm going to take a step back for a second and talk about some of the big scale stressors that are affecting coastal wetland ecosystems right now. So this is a map. This map shows areas in green, and I apologize for the kind of bad resolution there, but it's not my map, so not my fault, right? This is a USGS map that shows areas that are predominantly wetland and green. So immediately you can see from this map that we have high densities of wetlands up and down the East Coast. So today I'm going to focus on areas in North Carolina, and we're up here in Connecticut, areas in Connecticut. So if we think about what else is going on the East Coast, this is population change from 1970 to 2008. Pink means that population, or warm colors mean that population is increasing. If you look at the same area up and down the East Coast, you can see mostly warm colors, right? We have an increasing population on the East Coast over the past several decades. Now this is even more prominent in the Southeast. But if we look at population density, of course we all know we live in Connecticut, the population is really dense up here. We're right outside of New York City. Population density in the year 2000 per square mile is also quite high on the East Coast, especially in the Northeast. We're in dark blue up here in Connecticut, in the Long Island Sound watershed. So all of this population growth in these dense human populations means that we use land, and we use land for different things. The graph on the right shows percent development, so this is like percent urban land cover, or close to it. Now if you look, red is high, and anywhere that there is developed land is a color, so you see a lot of white. If you look up the East Coast, there is some development, and then of course New York City is that bright, red spot, the highest spot on the map. But you can see that we're quite blue up here in Connecticut. We have a lot of urban land cover. Now in contrast to this is areas of cropland fertilized as a percent of all cropland acreage. And what that is a measure of intensity of agriculture. So the darker green it is, the more intense the agriculture is, the more fertilizer we're applying. And so down here, so you notice, of course, right here, this is the Mississippi River Basin, right? The most intense agriculture in the United States. But if you look on the East Coast, we have some quite intensive agriculture here too, especially in the Southeast in these dark green areas. So along the East Coast, we have a lot of urban development in the Northeast, a lot of intense agriculture in the Southeast. Remember, I'm going to talk about those two different places. Okay, so what does this mean for wetlands? This is a cartoon that my master's student did for me. I really like it. She's good at cartoons. So this means that wetlands along the East Coast tend to experience really high pollutant loads because we all live around wetlands. And you can drive around Connecticut and see this, right? Wetlands are everywhere, and you can see them right in the middle of industrial complexes. And so we tend to have a lot of runoff with a lot of excess nutrients or contaminants like metals, either from agricultural operations or urban development. So we have this going on. At the same time, saltwater intrusion, which is just the movement of seawater inland, either through the groundwater or through the surface water, is increasing dramatically along the East Coast. This is a fairly old map just showing the saltwater intrusion, and as you would expect, it happens along all of our coastal areas, right? But we have a lot of saltwater intrusion along the East Coast. Now, over the long term, this is driven by sea level rise. We expect sea levels to rise across the East Coast. You'll notice a higher susceptibility south, and that's because it's much flatter. The coastal plain extends very far inland in the south, so you get higher susceptibilities, seawater moving farther inland. That's not to say we won't experience some severe effects of sea level rise here. Now, over the short term, saltwater intrusion can also be driven by things that are going on quickly and right now. One is the increased intensity and frequency of hurricanes. So storm surges push saltwater very far inland, right? So you get episodic saltwater intrusion events. And the second is the opposite of that, extreme droughts. When we have a drought, which are increasing at a particularly high rate in the Southeast, that lowers the water table and allows seawater to move farther inland. Okay. And so an example of this is here. So this is from a few sites that we've studied over the years. This is conductivity on the y-axis, so it's just a measure of salinity. And this is across the summer of 2007, which is one of the most extreme droughts on record in North Carolina. These are at 3 locations, moving inland from a sound. So this is the Scuppernong River, the Alligator River, and then the outflow of one of our study sites. And the first thing you notice is that, of course, all of these increase. But if you look at our study site here, which is a freshwater wetland, it starts out as freshwater, which is, 10 to a couple hundred microsemen per centimeter, right? Not even on this scale, really, really low, up to 12 millisemen per centimeter, which is higher than most brackish wetlands. So over the course of this drought, we ended up with a freshwater wetland that looked almost like an estuary. No effect at all on the biota, right? Okay. So, now what we have here are these systems that are experiencing this large amount of runoff from agricultural operations and urban development. And what I forgot to mention, and I'll mention here, I forgot to mention it because it's not in the cartoon, is that because agriculture and development have been around for a long time on the East Coast, most of these wetland soils have large legacy pools of contaminants, right? A lot of nitrogen and metals that have built up over decades and decades of runoff. So we have contemporary runoff and we have legacy runoff building up down here. And now we have a third factor to worry about. We have saltwater intrusion. Now we have saltwater intrusion coming onto these wetlands that are kind of have this buildup of contaminants. So what we're really interested in is understanding what happens when you get these two chemical fronts that kind of smack into each other, right? So those are the systems that we're interested in and the systems that I'm going to talk about today. Okay, so if we kind of revisit our conceptual model of biogeochemical cycling for this talk, kind of our simplified model. Now we're thinking about wetlands that have high contaminant loads, that have had historic high contaminant loads, that now experience sea salt intrusion. So we're adding a factor, an input factor, into this wetland. Oh, look, there was my colors. It was on a different slide than I remembered, but it still got messed up in the map conversion. That's okay. We're not going to focus on parts of different questions. We're focusing on 1 and 3 here for the remainder of the talk. Okay. Oh, I'm really good on time. I'll go slower. So I'm going to talk to you guys about 2 separate projects. One that is in its, at least for my part of the project, is in its ramping down stage. That's been going on for about a decade. And I'm going to give you a little background and some like super hot off the press results that don't make much sense to us yet. We just collected our first year of data on this project. So the first project is an NSF-funded project, and it has a very large group of collaborators. So it's all of us after a very, very rainy day in the field. Emily Bernhardt at Duke, Marcella Ardone at East Carolina University, Jen Morse at Portland State, Amy Bergen at University of Nebraska-Lincoln, and Jeff Poole, Rob Payne, and Clem Zurieta at Montana State, along with a lot of graduate students and research technicians over the years. So the setting of this research is on the coastal plain of North Carolina. So historically, the coastal plain of North Carolina was dominated by Pocosin swamps. So these huge cedar swamps dominated the landscape. Now during the 70s and 80s, we drained about 1 million acres of those swamps. 3/4 of those swamps were on the North Carolina coastal plain for intensive agricultural operations. So we turned this landscape into this landscape over the course of about a decade. And so to do this, all of the water is drained out, but the land is so low that it would just reflood. So they actually install mechanical pumps, and water has to be actively pumped. So pumps have to be maintained to actually keep the fields dry enough to farm. But the peat was so deep and the wetlands had built up so much, this topsoil was really fantastic for agriculture, at least for a couple decades. Then what happened was, like all across the U.S., most of our topsoil eroded, we lost a lot of it, and a lot of the agriculture became marginal. So active ag is kind of in this dark green color, marginal ag is in red. We have a lot of marginal agriculture. It's marginal because we've lost topsoil, so it takes a lot of fertilizer. A lot of fertilizer costs are associated with it, and a lot of costs are associated with keeping it dry enough to farm. So this creates this really interesting environment for a wetland ecologist because all this farmland gets abandoned. It's too expensive to farm, so people abandon it. So this opens up this really interesting opportunity for wetland restoration. So we're restoring a ton of these abandoned farms now. So the data that I'm going to present today comes from one particular very large kind of wetland complex restoration. It's a privately owned 10,000 hectare mitigation bank in North Carolina. The focus that I'm going to talk about today is a 440 hectare agricultural field that was formerly pumped, that was restored to a wetland about more than 10 years ago now, I guess. So this is what we call Timberlake, and I'll accidentally call it Timberlake, although officially it's Tower. It's the Timberlake Observatory for Wetland Restoration, but I'll call it Timberlake. So water flows this way. It flows off of upstream agricultural fields, and there's an unmapped inflow here off of upstream wetlands, and then there's one constrained outlet. It flows out of the site, through the Little Alligator River, and into the Albemarle Sound. So the site is freshwater. It should be freshwater. It's not necessarily always freshwater anymore. And it has wind-driven tides. So when the winds are right at the right time of the year, there is actually bidirectional flow at this outlet. So this is what the wetland looked like right before restoration, at least what part of it looked like. This is really interesting. This is a V-ditch here. So all of the farm fields in the southeast have V-ditches that drain water out. Very similar to the V-ditches. Has anybody ever seen the mosquito V-ditches in Connecticut? No, maybe? Very similar to the V-ditches that they created all over to Connecticut to drain wetlands to fight mosquitoes. And I don't remember what years they did that, but okay, so we planted thousands of trees, turned the pumps off, reflooded the wetland to its natural hydrology, and 5 years after restoration, this is what it looked like. Okay. And so these are some more pictures 5 years after restoration was completely complete. So the picture of the trees that I showed you is right here in the background now. And so these are just pictures from different vantage points. So it looks like a pretty well-developed, functioning wetland, right? So this is great. So we were spending all this time studying how wetland restoration affected nutrient retention capacity and greenhouse gas emissions, and then a postdoc on the project just happened to pull up the salinity data, and he was like, whoa, this isn't a freshwater wetland. So this graph shows elevation in water elevation in gray in the back, and then these are different sites within the larger site. And so you can just focus on the black. This is at the outflow, so it's at the location that's closest to the saltwater source. So you can see in these droughts, when the water elevation starts to drop, there's this immediate response in conductivity. Our measure for salinity here, and those are the wrong units. There's no cubed on that, by the way. So we get these kind of seasonal saltwater intrusion events at the site that we had no idea was happening. And remember, this 2008 to 2009 droughts were some of the most severe on record for the state of North Carolina. So these are droughts way outside of what we normally see. We had one more severe one during the project in 2011. So our question then was, well, what happens? What are the consequences for saltwater intrusion for wetland ecosystem function? So we have a site that looks something like this. We have agricultural runoff coming in upstream. We have a lot of legacy fertilizer built up. Remember, this was a farm field, an active farm field for decades. And now we know we have these large loads of sea salts coming in the downstream end of the site. So sea salts include large concentrations of anions like chloride and sulfate, cations like magnesium, potassium, and calcium. Right? So we're totally changing the chemistry of the system. So our first question, I'm going to talk about two sub-questions in this project. Our first question was, well, does the loading of sea salts affect how efficient this wetland is at retaining nitrogen? Does it change the amount of nitrogen that's exported off the site? And so this is great because the answer is actually really straightforward. These graphs show ammonium concentrations. Ammonium is one of the reactive forms of nitrogen. So we have ammonium concentrations on the y-axis and chloride concentrations on the x-axis. So chlorides are a measure of salinity. It's how salty the water got. For soil solution, so underground and surface water. And you can pretty easily see that there's a strong relationship for field and microcosm experiments and for field surface water collection, that as salinity increases, so does ammonium concentrations. So what happens is that the ions in the sea salts can actually kind of kick the ammonium off of soils, right? It can kick the nitrogen that's bound to soils off. So we get this big nitrogen release into waters that were formerly fresh, right? They're not used to that salinity. So if you've ever done lab work, like soils lab work, and done a KCl extraction, where you add potassium chloride to soils and all the nitrogen comes off, this 440 hectare site is like the giant KCl extraction. So in fact, we estimated that by the year 2100, saltwater intrusion into about 16 square kilometers of these kinds of wetlands with legacy fertilizers could release up to 18,000 megagrams of nitrogen, and that's approximately half of the annual ammonium flux to the Mississippi River, right? So this is a huge amount of nitrogen, and like I said before, we all live in the Long Island Sound watershed, so we know how sensitive estuary ecosystems are to nitrogen. So we asked this question about nitrogen export, and the second question we asked was, well, how does it influence the balance of greenhouse gas emissions? And so I'm going to talk about part of these results today. I'm just going to talk about nitrous oxide and methane emissions. Carbon dioxide emissions are roughly balanced on the site, we think. I'm not going to go there today. So I have to do just a little bit of redox chemistry. The resolution's not that great, but I think you guys can read it. I apologize. Just stick with me. I'm going to try to keep it as simple as I can. So hopefully everybody's seen a redox ladder. Yeah. Undergrads, I know everybody else has. So the way this works, remember I talked about oxygen being really important and the amount of oxygen there. So the way this redox ladder works is that oxygen is the most energy efficient way for microbes to respire. And when oxygen is depleted, there are other alternate sources, right? We call those alternate electron acceptors. So when there's no oxygen to breathe with, microbes can breathe with nitrate. When there's no nitrate, with manganese, iron, sulfate, this can be carbon dioxide or organic carbon. And each rung of the redox ladder is a little less energy efficient. So the way we typically think about it is one rung, so for example the reduction of sulfate, usually doesn't happen until most of the next rung is depleted, until most of the iron is depleted. That makes sense? So we can use this redox ladder to make what should be some really simple predictions about what happens with saltwater intrusion. So I also want to point out, remember our important greenhouse gases? So they're here with denitrification. Remember it produces N2, but also produces some nitrous oxide. And then here with methanogenesis, which produces all of the methane we see coming out of a wetland. So we know that saltwater intrusion and ag runoff add nitrogen. We're adding nitrogen, we would expect more denitrification. Remember there's not much oxygen anyway. So during saltwater intrusion and ag runoff, we would expect elevated fluxes of nitrous oxide. Now the opposite is true under saltwater and under elevated salinity under saltwater intrusion conditions. Saltwater intrusions add sulfate. And so we know sulfate reduction is a little bit more energy efficient than methanogenesis, right? So if we add a bunch of sulfate to the system, we would expect those sulfate reducers to outcompete methanogens for carbon that's available, and we would see less methane coming off of wetlands. So we see a decrease in methane. So we hypothesize to see an increase in nitrous oxide and a decrease in methane. Okay, so we tested this idea using a lot of different methods. For the sake of time, of course, I'm not going to talk about all of these. We did soil core experiments. We did field salt additions where we actually poured seawater onto wetland soils. And then we also did a field survey of greenhouse gases using floating chambers. My design, by the way, all duct tape, styrofoam, and PVC, like a real field ecologist. So I'm going to talk about the third just for the sake of time. So we did this survey across the site, across a natural gradient of saltwater inundation and agricultural runoff. We sampled these sites on 12 dates. All the sites were permanently inundated. So we don't have an oxygen gradient in question. We're in anoxic soils. And so this is what we found. So this is methane concentration. Fluxes show the same pattern. I'm just showing concentration for time and clarity here. This is methane concentrations across the gradient. These are our site numbers. So we have saltwater intrusion coming in here and agricultural runoff coming in here. And so you'll notice two patterns. One is that methane is only high in summer, which makes sense, right? It's warm, biotic activity is churning. So we have the highest methane concentrations in the summer. But across all seasons, methane concentrations remain low in the sites experiencing saltwater intrusion, right? They only get high where saltwater intrusion doesn't quite reach. The same as agricultural runoff. They remain low all year and only get high as we get less agricultural runoff, right? We have these two chemical fronts that haven't quite met in the middle. And so the reason for this is sulfate, of course, right? Our hypothesis. So we weren't surprised when we saw that pattern with saltwater intrusion. We expected to see this. We expected that sites experiencing saltwater intrusion would have very high saltwater, very high sulfate, particularly during summer saltwater intrusion events, right? So this makes sense. What surprised us a little more was that there was really high sulfate concentrations on the ag fields, which we didn't expect to see, but which actually made a really interesting pattern. What happened was that year the Mormon church actually owns the farms upfield from the stream. They had decided to apply a lot of gypsum to the field that year, so we ended up having a ton of sulfate runoff into the site that had never happened any years prior. So we have this very strong relationship with sulfate that explains a lot of variability in the data with sulfate alone, and an interesting gradient where we have sulfate with seawater and sulfate without seawater. And so we saw that sulfate suppresses methanogenesis, or at least the release of methane to the atmosphere, at a threshold of about 10 milligrams per liter, which is quite low. So this would suggest that saltwater intrusion reduces greenhouse gas emissions. Now remember, we thought the opposite was going to happen with nitrous oxide, and we were surprised to see that the opposite didn't happen. We see a very different pattern here. So this is nitrous oxide concentration across the site. And you'll see that it pretty much remains low almost all the time, barely above detection really, except for a couple of huge events in the fall, and these were big agricultural runoff events. They applied a lot of fertilizer and it rained immediately after, so we had a huge flush of nitrate onto the site. So our question was, so how do these two things combine? Right? What are kind of the combined effects of saltwater intrusion and agricultural runoff? Because we saw these two very distinct patterns with methane versus nitrous oxide, and we know that they have very different forcing factors, right? Methane is about 25 times stronger than CO2, and nitrous oxide is about 200 times stronger. So if we look at our data in that respect, the CO2 equivalents, so we make them equivalent according to their effect on the climate, we see that by exposing the wetland to seawater, we get this huge suppression of total carbon dioxide equivalents, right, of the effects on the climate. But when we add agricultural runoff to that mix, we see a big spike in nitrous oxide emissions. So these enhanced nitrous oxide emissions from agriculture could offset this kind of hidden bonus of sea level rise, right, of a suppression of methane emissions. So they kind of almost balance each other out. Okay, so we saw this big increase in nitrogen export. We saw a suppression of greenhouse gas emissions, but that suppression is questionable depending on the runoff coming into the system. So I'm going to give a very, very brief overview of this brand new project that we just started here in Connecticut. So this project is funded by Connecticut Sea Grants. We just collected our first year of data. Tim Vadas, he's a professor of engineering at the University of Connecticut, an assistant professor of engineering at the University of Connecticut, is a co-PI on this project. April Dorosky is a master's student in my lab who's done a bulk, or really all, of the work since the grant got funded, along with several undergraduate researchers, Mary Zawatsky, Christina Macklem, and Mary Scholl. And I bet Christina would not be that happy with me if she knew what picture I chose of her to put on a slide in front of a bunch of people. But that's okay. Okay, so in contrast to the highly agricultural landscape we just talked about, Connecticut's quite different, right? It's very developed. The gray in this map is urban land cover. The dark blue is surface water, and the light blue is wetlands. The black circles are Connecticut State wetland restoration projects. So the state of Connecticut has restored about 1,700, is it 1,200 or 1,700? Somewhere between 1,200 and 1,700 acres of wetland, of tidal wetlands in the past 25 years across 40 sites. So we have this urban landscape, and within this urban matrix we have very high density of wetlands, and we're really pushing to restore the wetlands that aren't hydrologically connected to the sound, right? So we're increasing the density of wetlands across this landscape. So wetland restoration is typically done in the state of Connecticut and in other places for habitat management and for wildlife, for things like birds and fish, and also to combat, especially here, invasive species. This is invasive Phragmites. A lot of wetland restoration in the state of Connecticut is done to combat this invasive. One of the primary means of restoring a wetland is a tidal flow restoration. And what this means is that they replace or remove culverts that are undersized, and or they install self-regulating tidal gates, and or they remove fill. And what all of this does is it restores the natural hydrology to the wetland, right? So the wetland is no longer cut off from downstream water sources, so now it has this nice natural hydrology, okay? And so this is also a good analog for sea level rise, right? We're re-inundating a site with salt water. So our question for this project is, how does urban runoff and sea level rise influence wetland functions in restored and natural wetlands? So for this project, we have 36 study locations across the Connecticut coast. They're shown here. The colors correspond to their salinity at high tide. So we have freshwater. So the wetlands are set up to be along a gradient of fresh, brackish, and saline wetlands. So we're covering the salinity gradient. They're set up to cover a development gradient, so wetlands that drain fairly undeveloped land cover, which is really hard to find on the Connecticut coast, to wetlands that drain very developed land cover. And then they're also divided into restored and unrestored wetlands. So wetlands that are natural, most of them are natural because they aren't in need of hydrologic restoration, right? Their hydrology is in its natural state. And then wetlands that have had some kind of hydrologic restoration. So we've done something to restore their natural hydrologic regime. So there are a lot of things we're thinking about under kind of this umbrella question. I'm going to talk about one very small one very, very shortly. This is what my master's students spent last summer doing. How does denitrification vary among restored versus natural wetlands along a salinity gradient? So just as a quick refresher, remember denitrification was that really important pathway because it's the only pathway that can take reactive nitrogen and transform it into dinitrogen gas in the atmosphere. So nitrate has a series of chemical processes. Nitrate's reduced to nitrite. Nitrite is reduced to nitrous oxide, where nitrous oxide is that important greenhouse gas. In both systems, the bulk of nitrous oxide goes through one other reaction, and it is reduced to dinitrogen gas into the atmosphere. So when we're thinking about the efficiency of nitrogen removal of a wetland, denitrification is the most critical process that we can consider. So, super quick results. Our first really surprising result, I meant to say something before I put that up there. pretend like you didn't see it. So we thought that this would be different for restored and natural wetlands in Connecticut. And the reason we thought this is because we assumed that restored wetlands would have been much more degraded over time, so would likely have very heavy contaminant loads in their soils. And we assumed because of all the fill removal and moving things around and being dry, they would have very different amounts and types of organic matter, right? Which microbes need. They have to have organic matter to reduce nitrate. But what we found was quite surprising because we found no difference, which is actually great, right? We found that restored wetlands and natural wetlands both have similar rates of denitrification. So this is potential denitrification here. This is a laboratory experiment. We went out and collected soil cores from all these wetlands and brought them back into the lab. For brackish, fresh, and saline wetlands, pink is natural and blue is restored. And so even though if you squint, it kind of looks like there's a pattern here, it's not significant. There's no significant difference between any of these categories. So what we're trying to do right now is to develop statistical models to try to figure out which chemical drivers are important for denitrification across all of these systems. This is a quick initial plot of denitrification versus soil electrical conductivity, which is our measure of salinity. If you squint really hard, it almost looks like it's decreasing, but not really. But when you run a multiple linear regression model selection, so you actually take into account some of the multiple factors in our data set, we do find a significant negative relationship with soil salinity and also a positive relationship with some of our metals. We don't really know why yet. There are a lot of complex interactions between the iron, manganese, and nitrogen cycles that I'm not going to get into today because I don't completely understand why our data are telling us this. But this is where we are right now, so brand new data. Don't quote me on it because it's hot off the press. So we're moving on with that project. Just for a super quick summary. So we're seeing, at least in what we've done so far, is that restored and unrestored wetlands have similar greenhouse gas emissions and denitrification rates, which is great for those that are really interested in restoring wetlands for wildlife habitat. There are added benefits to that. We saw that in both Connecticut and North Carolina. We saw that increases in salinity decrease greenhouse gas emissions in North Carolina, but this also really depends on contemporary pollutant loads. And we saw that increases in salinity increase nitrogen export through the mechanism of cation exchange in North Carolina. Remember the giant KCl extraction? And then by suppressed rates of denitrification in Connecticut. That's still a very initial result. I should have put that like shaded or something, right? We don't really know that yet. So I'm going to end on this slide. We're almost out of time. So we're continuing on in this research in our Connecticut project that's ramping up. We're really focusing on these different questions that tell us how saltwater intrusion affects wetlands within this urban matrix. We're starting to look at nitrogen and metal exports from these wetlands. We think metals might be very, very important. Legacy heavy metal buildup in urban wetlands. We're looking at how the microbial community actually mineralizes carbon and its capability of decomposing some of that organic matter. And we're looking at greenhouse gas emissions in these wetlands. And so we'll be doing that over the next couple of years. Okay, so I'm going to end there. And thank you guys for having me. It's really fun.
Thank you very much. Questions for Dr. Helton?
Is it possible to tell whether or not a wetland is, restored versus the natural wetland?
Oh, just by like looking at it?
Yeah.
No, not really. I mean, a lot of them, once their tidal hydrology is repaired, really grow up quite quickly. So the way we identified these wetlands was by working with Connecticut Deep. They actually have historic records of everything they've done at every wetland they've ever done it. They're not very organized, so we spent a few hours sitting in somebody's office like clicking through records trying to figure out which wetlands to select. But unless the restoration was like just done, they literally just went in and dug it out, after 5 or 6 years, I think it's usually very hard to tell, at least from what I've seen.
And then my second question is it known, I mean, for the wetlands you've been studying, do they know how much nitrogen fixation
No.
contributes to nitrogen?
No, that's a good question. So I Don't know the answer to that at all, and I don't even know any research that would point me in the right direction. What I can say is that cyanobacteria are quite rare, so I would guess at least algal nitrogen fixation would be pretty low. I don't know wetland in plant nitrogen fixers. Does anybody know?
They have legumes, rhizobia,
Yeah.
rhizobia, Freshwater.
So I don't know the answer to that, and I don't even know the research that would point me to that direction.
So how long does it actually take for a wetland to be restored?
It totally depends. So there are a lot of different kind of intensities of restoration. So the restoration I showed in North Carolina was like this gigantic planting. They had to let the trees sit there for 2 years because they didn't want the trees to get water stress. They had to remove these gigantic mechanical pumps, right? It was a ton of money. This huge real estate investment firm came in and did it so they could sell mitigation banking credits, right? So that restoration took like 4 or 5 years. It's crazy. Now some of the more simple restorations, like in Connecticut, what they'll do if they want to, there's a formal tidal wetland that has a lot of Phragmites. So sometimes the reason the Phragmites are there is because the water's too fresh. It's not really getting that tidal hydrology, the salt coming in. And so what they'll do is they'll just spray some herbicide, get rid of the Phragmites, and then pull out a culvert, right? And so that can be very, very quick. So there's like this huge range.
So when do you actually go in and do your denitrification studies? I mean, when do you decide, okay, now I'm going to go in after so much time or whatever, or things that occurred, now I'm going to actually do my measurements?
So for the first study, we did our measurements throughout the entire process. So I only talked about the ones after saltwater intrusion, but we actually did measurements from before to, they're still doing them now, to 10 years after the restoration, because it's that one study site. For Connecticut, what we've done is we tried to pick a range of ages. So we tried to pick a range of time since restoration, so we could kind of do a space for time substitution for the wetlands in Connecticut. We haven't actually looked at that data to figure out if the time since restoration is a significant predictor of something like denitrification, but that's on our list of analyses to do.
How, I guess, with the increase in saltwater to the wetlands, how halo tolerant are the plants or the primary producers that are in these wetlands?
That's a fantastic question, and it totally depends. So, something like a cypress is pretty intolerant to salt, but the cypress are still doing pretty well because it's kind of a seasonal event. So it depends on if the saltwater intrusion is constant, right, or if you're kind of getting these seasonal fluctuations of saltwater. So I don't know different plants and different salt stresses, but they do seem to do okay. Most plants do under something that is fluctuating relative to being constant. Connecticut has a lot of salt tolerant plants in the marshes, so Connecticut plants aren't as stressed. So one thing is, you asked about sulfide too, right? Or did I make that up in my head?
I think you made it up in your head.
Okay, good, because that's what I want to talk about. So they are stressed by salt. All plants also are stressed by sulfides, right? And so when you get all of this sulfate and all of this sulfate reduction, you get soils that start to turn sulfidic, which at least in our site seems to be the thing that really triggers plant death. Haven't thought about that as much in Connecticut, but in North Carolina that's definitely the case. The soil is starting to turn sulfidic, which is super interesting because it turns out that iron buffers how quickly soils become sulfidic. When you add sulfate to high iron soils, it forms pyrite, right? Iron sulfide. And once something is pyrite, it's super stable, right? It's iron sulfide for a long time. So the more iron pool there is, then the longer it takes for soils to become sulfidic. So there's a hypothesis that the effect of sea level rise on wetland plants could really be driven by the available iron pool in the soil. Whether or not that's true, I don't know. I just think it's a really cool hypothesis.
So you might be able to look at that in Connecticut.
Yes.
Because you've got a range of metal pollution.
Yep. So we've planned on looking at that in Connecticut, and we've actually talked to the NRCS about doing extra soil surveys. They're doing a new aqueous soil map and adding iron to their soil surveys, so we can try to get iron maps in coastal wetlands.
Just related to that, if we can come full circle on the acid mine drainage issues.
Sure.
Are there acid, I mean, I have no idea where mining occurs and where this is an issue. So your sites are you have sites in Connecticut.
We have sites.
Do we have?
There are legacy mining effluents in Connecticut. How much those interact with seawater, I have no idea. But pH is actually a really interesting question. So it turns out that seawater is more basic than freshwater usually is, right? And so with saltwater intrusion, you also get a change in pH, although the opposite direction of what you just asked, which if you think about about carbonate equilibrium, can have a big effect on the amount of carbon that's stored as bicarbonate versus the amount that's released as CO2. So that can actually have a big effect, the change in pH on greenhouse gas balance. I say it can. We think, right? It's a hypothesis. I don't really know the answer to that.
One more question.
Yeah.
One more question. My last question concerns infectious disease. So the salt intrusion obviously increases salinity, and so we know that there are infectious diseases, and this is probably more of a global problem, like Vibrio cholera.
Mm-hmm.
And so I was just wondering, I know they detected Vibrio cholera in, say, the Chesapeake Bay.
Yep.
But do you predict this, these kinds of things?
So I'm not a disease ecologist, so I have no right to make predictions. But I would suspect. I mean, so one example, this isn't a disease, but it's an animal. So there are specific mosquito species that are salt tolerant, right, that live in salt marshes. And so we're actually looking at some of our ephemeral wetlands that experience road salts for those species. And the reason we're looking for them is because they found those species in Michigan in ephemeral wetlands that have large salt loads. So, I mean, my guess is that things that are very salt tolerant, as soon as you start to increase the salinity, I don't know how long it'll take or when it'll happen, but will eventually move either with flow or with humans.
Any other questions? Okay. Well, let's thank Dr. Helton again.